topic called gastric secretion is one of the daddy topics, right? One of the biggest topics you can, learn, and it's amazing. It's amazing how our GIT works, and it just gives me goosebumps too. It's amazing. That's what I'm trying to get to. So the volume of, secretion is about 2.5. So our GI tract, it produces not produces, it just secretes around 2.5 liters of water inside a GIT, and it can get recycled. So that's the idea of 2.5 liter per day. The pH is mostly acidic, right? It's around 1.5 to 3. It's mostly acidic secretion, that is HCl. It produces mostly. And gastric glands, we have 6 type of gastric glands, not gland, 6 type of gastric cells. And this is how one gastric gland looks like. And here we can see it's a flask shaped gastric gland. Flask means test tube, so it's a test tube shape, if you can understand. Test tube shape, right? And it produces something called as HCl. Now HCl is amazing. HCl is made up of iron, H plus iron plus chloride. So remember that, because I will tell you why. So we have 6 cells. First is surface cells. Surface cells are of course present on the surface right here. They produce something called as bicarb rich mucus for the acid neutralization, of course. And we have something called as trifoil peptides, and they confers immunity. Now this term called trifoil peptides, right? They actually are the correct term here is not tri. The correct term here is trefoil, and they are called trefoil factor family, TFF peptides, and they are named in their 3 loops structure, okay? And they are stabilized by these bonds. Okay, what's happening with me? Okay, so members of these trefoil family, I told you, tri means tri means 3. There are 3. So there are 3 main trefoil peptides, right? And first is TFF1, second is TFF2, third is TFF3, right? And they are produced by their respective genes, TFF1, TFF2, TFF3 genes, right? And what happened here is that the main function of these is this, that they are the major sites of secretion, right? Of what? So TFF1 produce something called as gastric foveolar cells, right? And TFF2 are major producing site for gastric glands and these Burners gland, right? They are present at the duodenal, right? And the last TFF3, which is very important, is that goblet cells, and they produce a lot of mucus, and they are present in small and in large intestines. So we know 3: TFF1, TFF2, and TFF3. Okay, so that's the concept. The main reason, the main reason is that they confers immunity. It means they are cytoprotective. It means they protect our cells, right? And they are reparative molecules for the GI tract. And they don't digest anything. That's very important. They are just for the immunity. They are just for the protection of the lining, right? They protect the mucosa. They repair the mucosa. They act as a tight junction integrity. They maintain these tight junctions between our smooth cells, right? And if we have problems in these trifoil peptides, then our smooth junction will have problem in their tight junctions. And if we have problems in the tight junction, a lot of celiac, a lot of gluten can get inside these tight junction and can lead to autoimmunity mediated by these gluten, right? And we have something called as celiac disease, and then we will not be able to absorb enough iron, and then it can lead to iron deficiency, what's the name, this shape goes down. This basically we will have this high this ferric oxide, what is the name, iron deficiency anemia, right? Basically we will have anemia, right? Basically we will have malabsorption due to trifoil problems. Okay, so that was trifoil. Neck cells, they are present at the neck region. Now they produce something called as stem cells. They do not produce, they have stem cells, and they are the stem cells of the gastric gland. Now why they are not at the basal membrane right here, why they are not in the basement, why they are on the neck region? That's a great question to think about. Now, stomach has 2 different epithelial types, right? First is surface and second is glandular, right? Now, the surface type cells, right, the one we just read here. Now, these surface they are short-lived, around 2 to 3 days. And the glandular, the long-living ones, like right here, these are the glandular cells, parietal, ECL cells, chief cells, antral gastric cells. These are long-living cells, right? So we need these stem cells that are present in the neck region. We need the stem cells for this and this part, right? And this is short-living and this is long-living, right? And why? Why is it like that? Now it's a cool question. Now I'll tell you. If a stomach had stem cell at the base, they can only migrate upward, right? But the cell replacement has to happen upward and downward. So that's why the stem cells that are present in the stomach lining, they are in the neck cells, right? So that's like concept about it. I don't know why the hell my table squeaks a lot. I guess I'm gonna change it. So other question is that base is more acidic. That's why neck region is more safer for these stem cells. So they are near the mucus producing cell that are the surface cells. Okay, so we understand neck cells. Other thing is that parietal cells. Now parietal cells, always remember parietal with HCl. So parietal cells, they are living for weeks as they are less exposed, because these are, they produce acid, and acid due to gravity goes down, not upward, right? So they live for a lot of time. So they are less, and they produce HCl, right? And they produce something called as intrinsic factor, right? Now intrinsic factor is a glycoprotein, right? And it's a glycoprotein. It's an intrinsic factor for B12 absorption. Now I will tell you the story of B12 absorption, right? So B12, we have two type of B complexes, right? We have B12 and we have B9, right? So B12, we eat this in our mouth, right? It gets inside our mouth through the diet and it goes to the stomach, and our stomach releases HCl and pepsin, right? Two things. So both of these, they free the B12 from the food we ate, right? And now B12 is free. Now what happens is that here we produce something called as R protein from saliva, right? And this B12 goes and binds with this R protein, and then once it is binded with this R protein, it makes a complex, right? And why do we need to bind this? Because if B12 is not binded with R protein, it will be destroyed from the HCl. So B12 gets, inside our stomach, and it gets free from, this free from the food, right? And then it binds with R protein. Later this complex with R protein goes into into our duodenum, right? Now the story for intrinsic factor starts here. So now this B12 complex with R protein is in our duodenum, and intrinsic factor has entered the chats. Now this intrinsic factor goes to your duodenum, right? And what happened here is that pancreatic proteases, right? They are really strong, high. Protease means they destroy protein. So pancreatic protease, they digest the R protein. So again, the B12 is free now. Now what happens is that this free B12 binds with this intrinsic factor, because R protein was digested by pancreas. Now this complex of B12 and this factor, right? This factor called intrinsic factor, right? It has now, become, like, it is available now. Now this B12 intrinsic factor goes to ileum, right? And at the terminal ileum, via something called as Cuban receptors, this complex get endocytosed. Once it is endocytosed, it enters in the enterocytes, and then we need something called as transcobalamin 2. Now this transcobalamin 2 binds with B12, and after this, just B12 can travel into the blood. So that's like the story of B12 in short. So that's like one of the story. So we need intrinsic factor for the absorption of B12. So if we don't have intrinsic factor, if we have damaged parietal cells, we will not be able to make this B12 absorption. So we have something called as enterochromaffin like cells, ECL, right? These are not enterochromaffin cells, because enterochromaffin cells, entero means GI or intestine, chroma means color, and fin means they are colored. So these enterochromaffin cells, they produce something called as 5-HT, that is serotonin. And serotonin is a neurotransmitter. It's an excitatory neurotransmitter, right? So this enterochromaffin cells, they produce serotonin, but they are like, now these are not enterochromaffin, they are enterochromaffin like cells, okay? And they produce something called as histamine. Now histamine is also a neurotransmitter. Okay, now histamine goes and increases the acid production. How? Well, these enterochromaffin like cells, they are paracrine, paracrine gland, right? And they paracrine means there it is neither endocrine nor it's exocrine. So paracrine stimulator of acid, right? It goes, it stimulates, right? It gets secreted. Once the histamine is secreted, once it's outside, it goes and it diffuses with something called as parietal cells, right? So it goes right back to here, and once it stimulates the parietal cells, we know parietal cells produces intrinsic factor and HCl. So we have more HCl and we have more intrinsic factor in the end of the day. Okay, so here it diffuses with parietal cell and it binds with H2 receptors, right? Okay, so this was the whole, we'll talk about this H2 receptor soon. So we now understand parietal ECL cells. Now let's talk about chief cells. Now they are chief cells because they produce something called as pepsinogen. Now this pepsinogen is actually a zymogen, right? What is zymogen? Zymogen is just an inactive enzyme that wants to get activated. So this pepsinogen is very important for the digestion of proteins. Just remember that. Then we have enteral gastric cells. Now these enteral gastric cells, it means they are at the enteral part of the stomach, right? Now we know that stomach is like that, right? And the enteral is right there. So this enteral gastric glands, now they are G cells, right? And they produce something called as gastrin. Now gastrin is amazing. Gastrin is amazing because it increases the acid production. So whenever you see gastrin, think about acid production, right? What happened here is that G cell are generally kept under neural inhibition. They are generally, they are generally stopped. But once the inhibition is removed via the trigger of what? Amino acids, it means protein, vagal stimulation, right? But that vagal stimulation is not acetylcholine here, okay? Some other sort of vagal stimulation, okay? Or some stomach distension. Stomach distension means your stomach is getting distended because you ate something. So this whole of these, all three triad leads to acid gastrin, which is this product, right? Gastrin binds to something called as CCKB. Now these CCKB cells that are present on parietal cells, right, or ECL cells. Okay, so once the CCKB binds to gastrin, now this complex goes and it activates something called as GQ coupled protein receptors, right? And once this complex is activated, we have positive effect of H and K ATPase. Now this pump called H plus and K plus ATPase is responsible for the formation of H gives us H, and we need, okay, this usually what happens is that this HK ATPases make acid. Just remember that right now, okay? We'll talk about this whole thing later on because we have this acid production on our way. Okay, so note that in pernicious anemia, we have autobodies towards our parietal cells. So our parietal cells are cooked. Once we have bad parietal cells, we will not be able to make acid, which will lead to increased population of bacteria and which will lead to bad intrinsic factor. If we have bad intrinsic factor, we will have less absorption of B12. So what happens? Restriction of parietal cells, no acid. It's called achlorohydria. It means no acid. Or we have something called as atrophic gastric mucosa. Why? Because we have less stimulation of these parietal cells. Now our size of the stomach will, the mucosa, not the size, the mucosa that produces the parietal cell will atrophy, and then we have anemia because B12 is very important for the production of hemoglobin, right? Not hemoglobin, RBCs. So that's like the concept right there. So pernicious anemia, we have achlorohydria and anemia. So that's important. Okay.